The data are available from DRA, DDBJ Sequence Grid Archive. You can search the data from DRS search page by accession number, organism name, and so on. This time, we will obtain this data registered under the accession number shown here. It is derived from whole genome sequencing of Lactobacillus hokkaidoensis, with an estimated genome size of 2.5 megabase pair. Let's find this data. Enter the accession number here, and then press the search button. Now you get the result here. For more details of this data, please navigate to experiment page from here. This page shows how this data were obtained. It is obtained using Illumina MySeq sequencer and using paired end sequencing. Paired end sequencing means that the both ends of DNA fragments are sequenced. So this data contains two files. One is for forward read or read one, and the other is for reverse read or read two. This DNA fragment to be sequenced is called insert. This data has an average insert length of 520 base pair, and each read has read length of 251 base pair. To download the files, let's go to the download page from here. Now you can see two files. One is for read one and the other is for read two. Before downloading these files, let's make a working directory for this lesson. Make directory analysis and change directory. Right click here and copy the link. Then go back to the terminal and download the file using curl command. And paste the link here. Now I have downloaded the file for read one, and next download the file for read two. Change file name like this, and then press enter. Okay, now I got two files. Next, let's unarchive these files. The file format is BZ2 file. So to unarchive these files, use bunzip2 command. You can use asterisks as a wildcard. So by using this, you can process all the files that end with the extension .bz2. This may take some time, so let's wait patiently. Now it's done. Let's check the results. There are two new files with a file extension of .fastq. Let's see statistics using seqkit command. Each file contains about 3 million read sequences with a read length of 251 base pair. The file format is fastq. So let me explain this file format in the next slide. Fastq format is a standard format for output from high-throughput DNA sequencer. Each sequence consists of 4 lines. In this example, this part represents one sequence data. First line for sequence identifier, second line for low nucleotide sequence, and fourth line for quality score.